Okay, great. So I'm going to walk everyone through some updates in terms of it's kind of the last part of the lecture today. So I'm going to go through this a bit quickly, but give you an update on how we kind of got to where we are and some of the RNA sequencing methods that are old school, some more recent work, which you've just heard about from Dr. Horner, and also a little bit of RNA viruses work from Dr. Tierney, and walk you through some of the other work that's related. So now, as most of you know, Jim Watson and others in Crick put together kind of the central dogma of molecular biology, this very simplified unidirectional sort of framework for what we think about for how information flows in biological systems. Of course, there's many variations of this, even that Crick put forward, but this is kind of the simplest central dogma a lot of us think about often. But then, of course, we talked about a couple of weeks ago the epigenome, the regulatory layer behind DNA, how DNA is packaged, modified, controlled, and then leads to its sort of elucidation and sort of new functions, or how it basically creates new phenotypes. So we did a lot of this, a few weeks ago, and this was because there are small chemical tweaks that can confer, new phenotypes, new regulatory control. So again, a bit of a refresher of what we already talked about a couple of weeks ago. But I'm going to come back to this because a lot of the same questions show up again in RNA. And so in particular, if you remember, what came up on the slide a couple of weeks ago is that, methylation marks the spot. There's gene boundaries. This is where you actually see the dip in DNA methylation, and they can tell you in normal cells versus cancers how it was perturbed. And so again, this is from a couple weeks ago, but thinking about this now, if you think about, where we find genes, it kind of begs this question of, well, how many genes are there? This is actually a question I've had ever since I was a grad student, and I'm woefully much older, but the question is to some degree is really the same, is how many genes are there in the human genome? What actually are there? And so I did an original review back in 2009 of how many genes. Well, there's, about 2% of the genome is exonic. A lot of the genome is introns. A lot of it's transposable elements or segmental duplication, structural repeats, and these even break down by different classes of repetitive elements and transposable elements. And so a lot of the, sections of the genome, how many genes there are, depends on where you look. You can see here there's a lot of areas of the genome that you would think are not, well, a lot of people call it, they used to be called junk DNA, which makes some people break out in hives at certain RNA conferences because calling something junky just means you don't understand what it is, which seems uninventive at the very least and misassigned at sometimes at best. And so when I was actually grad student and postdoc, one of the first plots I made of is an IGV plot, actually, and then translated into a UCSC genome browser. We were doing RNA sequencing of a gene called SLITTRAC1, implicated in syndrome. And I was very excited because some of the very first data I got, I took RNA-seq data, we aligned it. We've talked a bit about alignments in the past few weeks. But you can see this is the expression levels up to 10x coverage across these different regions of an adult and fetal human brain. And I thought, great, we're seeing exactly where SLITTRAC1 is. This is what we expect. It's a single exon gene, small 5' UTR, small 3' UTR, very simple gene. And I was like, okay, great, we found that gene. But then as I started zooming out, I started seeing other bursts of gene expression, these sort of non-coding RNAs that let's see, because here's the coding RNA, the exon, the coding sequences. But there's clearly some other expression nearby this gene, something else going on. And then you kind of zoom out a little bit more, you could even see other areas. Like this whole actually region was pretty transcribed, and not in all the tissues, but more in some versus others, like the temporal lobe more so than the amygdala or other tissues, at least in the fetal time point. So it's really got me intrigued back then, and even still to this day, of like this, it's 3 billion letters of reference of DNA, but how much of it's actually expressed? And part of the reason I was curious about it is because some of this even come back in my PhD work, which was, if you look at we took probes all across the genome for Drosophila, and we threw some non-exon probes, just threw them on the array, thinking, well, these are good negative controls. But it turned out half of them were expressed, which also the main exciting points of my thesis work was like, well, that's weird. What are they doing expressed? I thought they'd just be non-coding areas, but it turns out they are expressed, as we've known now for 20 years, at least in Drosophila, and eventually in yeast, and then also in other species, humans or Arabidopsis has been shown in many others. And so this gets to the point of where we are today, which is interesting. So there's non-coding genes, there's coding genes. And if you look at the number of coding genes, that number is 20,000 today. So you might get interviewed by someone like a reporter, could be your family, you could be at a Christmas dinner, and your grandma will say, oh, you're studying genes. What genes? And they might say, well, I'm studying this SLITTRAC1, or it could be any gene, could be IDH1. And then someone will say, oh, yeah, aren't there how many genes are there? And if any of you ever say there's 20,000 genes, I have the right to tackle you and attack you in a violent way, because if you say there's 20,000 protein coding genes, that's very true. But if you just say genes, that's not true. There's at this point, this one I published just a couple of years ago, there's, over 60,000 annotated genes, which includes protein coding, long non-coding RNAs, some pseudogenes, some of them also in repetitive elements, and non-coding RNAs here. But there are a lot of genes. And in particular, Just yesterday I was updating the slides, and this is where we're at today. So GenCode genes is the most well-annotated, curated set of genes. We're currently over 63,000 annotated genes in the human genome. And you can see this is kind of an amalgam human of many ancestries and backgrounds, which is better. You should just be just a sort of sad-looking older white dude. Now it's an amalgam creature on the upper left. But you can see here, there are still actually, around 20,000 protein coding genes. But, some are considered read-through genes, where essentially RNA polymerase II has kind of gone through, but there's still a lot of these non-coding. And there's actually now more non-coding genes than coding genes, which still upset some people in the field because they think maybe only a few of them are active. If you ask an X chromosome that's being silenced by Xs, for example, it will agree that some of them are definitely functional, or hot A or other non-coding RNAs. But I'm not here to belabor non-coding RNAs today. That's a different lecture, but there are a lot of them. But I do want to point out some other facets of RNA sequencing that have been useful that then blends into some of the direct RNA sequencing. And one of them is one of my just favorite facts about the transcriptomes potential complexity. So if you look at, if you have one exon, you can make kind of one thing. If you have two exons, you can make three variants of them. You can three transcripts. If you have three exons, you can make 7 transcripts and make 3 splice junctions. And it goes up at about this rate. And eventually you get to 2 to the n minus 1 of the number of possible variants of isoforms you could make. And the number of splice junctions is n times n minus 1 over 2. And what I like about this is because the human genome has so many genes with so many exons, including Titin, for example, actually there's more theoretical transcript combinations than there are atoms in the universe, assuming some basic assumptions about the universe. So it means that even if you wanted to, you could by definition not make all the transcripts that are possible in one of your cells because, well, there's not enough atoms in the universe. And of course, transcripts are made of more than even one atom. So this is, a fascinating concept of what's in each one of your cells. And so jumping back to when I was a postdoc and grad student, I was like, well, we need a way to sequence them all. So those are the early days of RNA sequencing. First started in 2008. Thinking like, how did we get to the RNA sequencing realm? A lot of it was, this was one of the first papers by Marta Zarve and Barbara Wold. Who's actually now contributed a lot to the STAR aligner, which you might use for some of the homeworks. And this was where we could start to look at poly selection. You shatter the RNA. You just heard Dr. Hunter talk about how it's very sad to take beautiful, intact, long RNA and then you shatter it with chemical elements. But one of the main reasons you do that is you get higher yield of cDNA when you do that. So we'll talk about, we've heard about today and we'll talk about more is direct RNA sequencing where you don't have to do this, which in principle should let you reveal more about the native state of that molecule. And then you map these tags onto the genome. At the time, it was 25 base pair tags back in 2008. And then you make what's RPCam, which I'll come back to that, and this let you then map across the whole genome like I was first doing as well as a postdoc. One of the first questions we looked at is, could we actually just replace microarrays? And I made this sort of tug of war slide. This is as good as my artistic ability gets. This is why I became a geneticist and not an artist. But anyway, the idea to see, well, could we just, at the time, everyone was using microarrays, and could RNA sequencing replace it? And so one of the questions is for, if you just say, well, are there differentially expressed genes? This is still something, a big part of your homework, and also something that comes up today is, in the early days, people would just say, well, a twofold fold change. Something that's 2 times different and bigger, we'll call that different, but doesn't take into account variance or small changes. So you have to include a measure of statistical significance as well, which is now, of course, standard, but you probably would be shocked to know that there was a time when people just did a 2-fold cutoff and then published that and said, okay, voila, we found things that are different. And when we first started looking at genotyping arrays versus RNA sequencing, it's RNA-seq up here on the y-axis and arrays on the x-axis. You can see for the most part in red are the places with high coverage, mostly correlated, you could see along this axis, but there were a lot of genes that you could see on one platform versus another. And we've done a lot of cross-platform comparisons. I'm not going to go into great detail today, but this is true for literally every single technology. There's no such thing as a gold standard where one platform can see them all. There's no one ring to rule them all. There is an eye of Sauron that is disrupting your quantification no matter what you do. And this is true everywhere we've ever looked, but there is generally correlation. Even you just hear from Dr. Horner's lecture, most things most of the time will agree, but in every case, there are some things you can only see well or only see at all with one technology versus another, which you can see here on these axes that you don't see any differential change, but you see a lot on one platform. In any case, what we could see, if you look at differentially expressed genes, we could see most of them would agree, but there were some that were unique to the sequencers versus the arrays. Ended up calling the title for this working paper was the death knell of microarrays, which was rejected by the editors. So it ended up being called instead an assessment of technical reproducibility in comparison with gene expression arrays. The death knell did not make it through the editorial process of genome research. But then the big question that came up in the field, and this comes up today, and this will be part of your homework, is, okay, you want to sequence a transcriptome. You'd like to know what's there, how many molecules, how complex it is. It depends on a lot of factors, at least these, which includes how long are your reads. And we'll talk about that for Nanopore because now they're much longer than they used to be. They're no longer 25 bases. They're now tens of thousands or thousands of bases. It depends on the size of your transcriptome, how complex, how much splicing is there, how much heterogeneity do you have in your tissue, biological variants, also errors. And so one of the first papers that looked at this question was, okay, let's say, for example, if you're just yeast, how many reads would you need here, and how many orths are you detecting if you have 4 or 5 million reads, or if you have, do you need tens of millions of reads? You can see here this was a kind of a debate in the field early on. But you can already tell here yeast is a much smaller genome than say mouse. So it's looking in the megabases versus gigabases, right? So you think about it's a smaller genome, smaller transcriptome, and this you can see this is at 9 million reads. You're already at plateau, and here at 9 million reads, you're just getting going basically. So different complexity transcriptomes require greater depth. And the way we also quantify genes, so again, it'll be part of the homework and part of the discussion is we basically would take all the reads and you want to normalize for how big is your gene. Because if you have something that's 10 kb versus 1 kb, if you fragment that molecule, you've got 10 times more chances to see it. Versus also how deeply you sequence. If you take marbles out of a barrel and you sequence a million marbles versus 100,000, you'll have of course 10 times more chance to see rare transcripts or rare marbles. So this was the original proposal for how to create basically reads per kilobase per million reads, or FPKM is when you have paired end data. And this is a metric that you'll see in the literature and you'll see a bit in some of the work we're doing. But then there's some other variations of this about, FPKM I just told you, but also transcripts per million reads is that you divide the read counts by the lengths of each gene in kilobases. And then you basically then create a per million scaling factor, and you divide then these RPK values by the per million scaling factor to get TPM. So there's a nice blog here, for example, but these are slight variations of a way to normalize for how deeply did you sequence and how big is a transcript and this complexity question. So you'll see this in the literature. You'll see this a bit in some of the homework, but this is its calculations relatively straightforward. You can see here, and the goal is to normalize mostly for these two things. So generally, TPM normalizes a little bit better across replicates. You can see here, depending on how big your gene is, and if you look at your replicates and look at, say, if you have a gene that wasn't really present at all, you can see here that just the variation in RPKM is a bit more so than say TPM, but they're not too dramatically different. But generally, people move mostly to TPM based on sort of this idea of how consistent you can get your data for the final metrics across replicates. Now, so that's kind of some of the numbers and the controls you'll see. That's the transcriptome. So I want to do a quick 10-minute summary of how do we get to RNA sequencing from the very early days of really 2007 and 8 when it first kind of was invented. Invented. There was cDNA and ES-based sequencing in the 90s, but basically the first time you could do it at scale with millions of reads. Actually, I took out the slide, but last night I originally had to talk that in the early days of Selecta sequencing, every run of the machine would generate more data for whatever tissue you picked than had ever been made before. So of course you didn't care that the machine took 12 days to finish, because when it got done, you said, I have just in one experiment generated more data on brain transcripts than have ever been made before, which was fabulous, which is some of the data I just showed you. Okay, so that's a transcriptome. What about the epitranscriptome? Now, you've heard a little bit about this already from both lectures before, especially Dr. Hunter. And just like, the genome has many chemical tweaks to think about, there are many RNA modifications as well. So Chuan He is one from his review papers. There's 6-methyladenosine, variations of 5 or 1-methyladenosine. You can see alpha-ketoglutarate also can create modifications. So this was earlier paper that was trying to figure out, we know there's variations, but what are they all doing? How many are there? Where are they? And so fundamentally, the question's about the same. As we have a methyl group or a small chemical tweak, and what does it do? What's the function of it? What is it actually mediating in terms of a phenotype or a cellular regulation? Some of which you heard about, especially in the context of viral regulation. But some of the papers you just saw cited included this one, which was, we were doing some of the very early first work here at Cornell, which was, okay, we have M6A and RNA. We think we can see it with an antibody. Does it appear in different tissues? Yes, it does. You can see here across the band. It occurs more across all of them. Does it appear more in adult versus embryonic stages of mice? More so in adult, but definitely present in all of them. You can see the intensity and also across multiple cell lines. So that, okay, it's definitely present across multiple tissues and stages. This is definitely not just a fluke kind of modification. But then the method that we invented to track this was MeRIP-seq, which you can see here is basically, again, fragment the RNA, take this beautiful RNA and you fragment it, but then you want to pull down the RNA elute, and then you can make these peaks after you do the sequencing, which you've already seen a fair amount of in the previous lecture, so I'll go through this quickly. But we saw this canonical enrichment at the end of the coding sequence, beginning of the 3' UTR. I also saw it in human and mouse samples. And so we knew that this was definitely something that was consistently showing up and had probably some selective constraint because it would show up across multiple species. And so this is kind of exciting. And, you know, first time, also fun fact about this paper, the first time I made this plot here, it worked in the second experiment we ran. It got all flat because the IP didn't work as well. So I thought, oh, maybe it's all noise. And for a few weeks, I thought, oh, maybe it's all noise. Maybe we should probably do the experiment again. So if you do an experiment and you've only done it twice, you should probably try it a third time if you think there's something interesting in your experiment, because we sat on this for a few weeks thinking that the IP didn't work or the antibody was bad. But it turned out to be fine. So then this started in 2012, this ability to globally map RNA modifications. I like to call it the birth of the Epitranscriptome, a bit dramatic, but I think it's definitely what it was. And particularly, you know, this is the same way we think about DNA being modified and regulated and moved between these different chemical states. The same thing is true of RNA. You can modify it and tweak it in different states to basically have it move between what states could be degraded, sequestered, or be used essentially sometimes as a way to cloak something in terms of a virus, for example, which you just heard about. And so today, at least as of last night, there are now over 400 RNA modifications known. This is a slide I modified from Nadia, but you can see, you know, a lot of them are in tRNAs, but it can function without modifications. And bacterial tRNAs and ribosomal RNAs have much less modifications, significantly less. And bacterial mRNA so far really has no known RNA modifications. And as you heard, also, it's also the distinction between viral and bacterial and mammalian RNA. And if you ever wonder, are there any other mods? Where are they? There is something called ModOmics database, which is kind of a play off our 2012 Epitranscriptome paper in terms of the layout here, but it's really, you can see there in eukaryotes, archaea, eubacteria, the different kinds of RNAs. There's also something called undefined RNA, which I think is fascinating. All these other weird RNA types that aren't quite defined. Again, many types of RNAs, snow RNAs, small nucleolar RNAs, peewee RNAs, many kinds of RNAs, and across many species. So they're everywhere, much like DNA modifications, and there's many of them. But the one thing we've seen, and again, because you heard a little bit about this in the previous lecture show, I'll mention it briefly, is that the MeReep-seq method, while we loved it and it helped us find a lot of biology, one of the biggest challenges, it's an IP-based method. And so you have to require ideally 5 or 6 replicates before you start to get consistent detection of any IP. So this is why it's kind of like there's an old saying that every writer has to kill one of their loved ones every morning. An idea that is not an actual person, but you should kill your darlings if you want to be a good writer. At some point, the methods that you help create or build may not be the best. So you have to always be looking to improve them. In this case, MeReep-seq is great if you have 7 or 8 replicates, but most people don't have 7 or 8 replicates, either time, money, or even the RNA input. And so again, some of the methods you just heard about is, you know, there's many methods that you just saw, and including bisulfite, glory-seq, or praise. But ideally, Nanopore could let you see everything. You see all the modifications, you get phasing of the variants. You could actually, you know, also look for poly-A site differences. In theory, this is what we'd love to see more of. And we're not the only ones. There are people just saying a call. I've never written a paper quite like this, although I guess I did write the birth of the Epitranscriptome. But that's just a call for direct sequencing of all RNAs, which I really love because I love the idea, but it's more expensive. And the field was not, totally frank, was not quite ready over the past few years. This is the Prometheon, which you've already seen, but you'll see more of it in the next week in lab. And we start to run some more samples. And I think what's really extraordinary is for direct RNA sequencing, again, not cDNA sequencing, which you can also do, but for the native, naked direct RNA all by itself going through the pore, as you can see here, let me go back here. You're actually pulling you make first strand synthesis, cDNA goes out to the side, the native RNA itself goes through the pore. So when you're doing that, over the years, it's gotten, you know, used to be 15% error, maybe 10% error. But just in the past, really, you know, really, well, say 8-9 months, it's gotten now to 5 or 6% error. And you can see that some are lower alignment quality, but some higher. And so it has really changed from in 2019, like, yeah, you could directly sequence RNA, but it was full of errors. Those colors are different errors aligned to the mitochondrial genome. But these days, it's actually much better. And actually data that you'll analyze partly for the class and that we've seen recently is we're getting, you know, again, 5 or 6% error, but getting really long fragments. So fragments 1, 2, 3 kb of complete native RNA. So I think it's just fascinating because it's been a long time brewing. It's been, I don't know, 20 years brewing, I feel like. It's something I've been thinking about ever since I was a grad student. But it's finally here, I would say. Like we actually now can do native RNA sequencing, and 5% error rate, you know, is not perfect, but it is. But because the molecules are so long, mapping them is not really the problem. You know exactly where they came from because there's so much information in such a long molecule. And so you can also sequence over multiple times to get enough depth to characterize it. And so one other fun fact about the sequencing is that you can do thousands of samples if you want and get it down to maybe $730 a sample if you happen to have like almost $1 million sitting around. If you do happen to have $1 million sitting around, we should just come see me after class. We should have a lot of conversations. But one of the things you could do is you could buy cheaper flow cells and library prep kits and do it at scale. But this used to be 1,000 or 1,500 a sample, so it's coming down. It's still not cheap, but it is something that you can start to actually do. You couldn't do it really on any other platform, so it's actually kind of exciting. And what we know about it is it actually does have an impact. So, for example, the RNA demethylase LPH5 impacts RNA metabolism, mouse fertility. So, for example, we want to find better ways to keep sequencing it because we know that if you have it or don't have it, in this case, if you don't have it, actually literally disrupts spermatogenesis, testicle sizes got smaller. You can see here that actually would change sperm count, even the mobility of sperms. This is just another highlight of that. There's many papers showing that the RNA modifications are not just decorative, they're functional. They will change literally your ability to reproduce or change a lot of functions and facets of cancer biology. And you just heard from Dr. Horner too, it's starting to creep its way into clinical trials, which also is very exciting because 10 years ago, we thought this was interesting and intriguing, but now it's actually something that's being targeted and deployed for clinical questions. And one of my favorite things about this paper that I included here is that you think about M6A in RNA, but then you look at what are the other genes that are affected. Say if you look at this ratio change, or you look at what's the fold change, here we go, going to fold change, what other genes are modified? You'll see DNA methyltransferase. You'll see other epigenetic modifiers for DNA methylation. So actually, I think there's an axis between the epitranscriptome and the epigenome that they're constantly in play and regulating each other that you can't forget about. And also, one last kind of fun fact about RNA is that work from Oded Rachavi, who will probably be visiting hopefully later this summer to give a talk here, has done transgenerational antiviral RNA response. So in C. elegans, at least, you can actually have a memory that's transmitted in the RNAs across several generations that they've seen a virus, and then they transmit that information to their progeny. So it means that even there's an inheritability component potentially to RNAs. And so if we go back now to the central dogma where we started, it really is not this simple. Of course, most there's many kinds of RNAs that guide how DNA becomes RNA or how you can actually translate, of course, RNA into protein. There are DNA transposons, DNA segments that can copy themselves and make copies and then transmit them. There's reverse transcriptases, RNA binding proteins, like you heard about ApoBex or ADARs that can actually go in and modify RNAs of the proteins. They go and modify them and change the information content. There's ribozymes that can also copy themselves. There's many variations of small RNAs as well here. There's prions, which kind of freak me out. I don't know if you're freaked out by prions, but like maybe I shouldn't eat meat because I don't want to get mad cow disease, which is creepy because they're just a protein that touches another protein and it replicates. But the creepiness factor of the central dogma is that actually at all levels, ribozymes, DNA transposons. So I felt a little better about the creepiness of prions after I made this figure, basically. And there's the epigenome, the regulatory layer behind DNA, the epitranscriptome, which you've not heard about today and last week. And in theory, we should call all post-translational modifications the epiproteome. Nobody uses that word. Everyone just says PTMs, but it is a realm of regulatory layer of the protein where you don't change the amino acid sequence, but in theory, change its function. And so this is something that it does appear in PubMed, I think 5 times. So some people have said it, but it hasn't really caught on as much as epigenome or epitranscriptome. There was actually a guy, Owen Birney, who's the head of EMBL. When epitranscriptome first started appearing in the literature, he was like, what is this word? This word doesn't make any sense. He's sort of come around, but it took like 10 years. There's also inherited DNA. You can actually have DNA information potentially epigenetically as remnants, for example, of trauma or starvation has been implicated in a number of studies. Definitely in mice, probably somewhat in humans. As I just showed you, viral-induced RNA responses are transmittable between generations for worms, which is interesting. And also prions can even go between species and generations, and it's kind of creepy still. But this is, I like to say, the updated central dogma of which you just heard about today. Questions?